Welcome to our podcast on microfluidics. Today we're going to start by breaking down the basics and then move on to a more critical debate of the field, enriched with real-world examples and analogies. I'm joined by Dr. Samantha Reeves, a leading researcher in microfluidics. Thank you for joining us today.
Thank you for having me. I'm excited to discuss microfluidics in depth.
Let's begin with the fundamentals. For listeners who may be new to the subject, can you explain what microfluidics is and why controlling fluids at such a small scale is important?
Microfluidics is the science and technology focused on the manipulation of fluids within channels that are on the scale of tens to hundreds of micrometers. At this scale, the behavior of fluids is dictated by forces that are not as significant in our everyday experience. For instance, viscous forces dominate over inertial forces, resulting in smooth, predictable flows, what we call laminar flow. This predictable behavior is a key advantage because it allows us to design systems where the flow and mixing of fluids can be controlled with very high precision. In practical terms, microfluidics enables us to perform chemical reactions, biological assays, and diagnostics in volumes that are drastically smaller than conventional methods require, which translates into faster results, lower reagent consumption, and often lower costs.
That sounds incredibly useful, especially in applications like medical diagnostics and chemical synthesis. How do these microscale principles translate into real-world benefits?
The real-world benefits are significant. For example, consider point-of-care diagnostics. A lab-on-a-chip device, which is essentially a microfluidic system, can analyze a tiny drop of blood to detect various biomarkers rapidly. This is extremely valuable in clinical settings where quick decisions are needed. In chemical synthesis, microfluidic reactors provide exceptional control over reaction conditions, temperature, concentration, and mixing, leading to highly reproducible outcomes. This is particularly important when synthesizing materials like nanoparticles, where uniformity in size and shape can make a huge difference in the material's performance.
It seems the control you get at this scale offers a level of precision that's hard to match with traditional methods. But let's talk about the basics a bit more. Can you describe how the fluid behavior in microchannels differs from what we see in everyday large-scale systems?
In everyday systems, such as water flowing through a garden hose, the flow is typically turbulent. That is, the fluid particles move in chaotic and unpredictable patterns, which actually helps with mixing. In contrast, in microfluidic channels, the small dimensions ensure that the Reynolds number, the ratio of inertial forces to viscous forces, is very low. This means that the fluid flows in a very orderly, laminar fashion. Because there is little to no turbulence, the primary mechanism for mixing in these channels is diffusion. Diffusion, while effective over short distances, is much slower than turbulent mixing. That's why engineers have developed clever channel designs like staggered herringbone mixers or serpentine channels to increase the interfacial area between different fluids, enhancing mixing even in a laminar regime.
It almost sounds like working with microfluidics is like managing a very organized, controlled flow of traffic rather than the chaotic rush hour scene we're used to. Now, transitioning from the basics to more critical issues, some critics argue that microfluidics is overhyped and that its practical applications remain limited. What is your take on these critiques?
It's a provocative debate. Early microfluidic devices were indeed seen as laboratory curiosities with limited commercial application. However, over the years, the technology has matured considerably. We've seen substantial advancements in both materials and design techniques that have extended the range of applications. That said, there are valid criticisms. For instance, issues like scalability and reproducibility still pose challenges. Many microfluidic devices work well in controlled lab conditions, but transitioning these systems to robust industrial-scale applications requires overcoming several hurdles. Despite these challenges, the progress made in point-of-care diagnostics, chemical synthesis, and even environmental monitoring suggests that microfluidics offers real and practical benefits.
Let's drill down into that scalability issue. Many companies are hesitant to invest in microfluidic technology because of concerns over consistent performance across large-scale production. How do you respond to the claim that if microfluidics can't be scaled reliably, it will remain a niche technology.
Scalability is indeed one of the toughest challenges we face. Early devices were often handcrafted and tailored for specific experiments, making batch-to-batch reproducibility a significant concern. However, recent advances in fabrication methods, such as soft lithography, injection molding, and even high-resolution 3D printing, are beginning to address these concerns. Researchers are now focusing on modular designs that allow for standardization, which is key to scaling up production. The development of standardized components and improved quality control measures is making microfluidics more robust for industrial applications. While the road ahead is still long, the trend is encouraging.
Let's consider the dynamics of mixing again. You mentioned that because the flow is laminar, mixing relies on diffusion, a slow process. Critics say that this is a fundamental flaw in microfluidic design, especially for time-sensitive applications like clinical diagnostics. How do you justify using a process that might inherently slow down mixing?
That's an excellent question. While it's true that diffusion alone can be slow, microfluidic engineers have developed ingenious solutions to overcome this limitation. One popular approach is the design of staggered herringbone mixers. These involve creating grooves on the surface of the channel that force the fluid to fold and stretch, effectively increasing the contact area between different fluid streams. Another approach is to use serpentine channels, where the winding path forces the layers of fluid to repeatedly come into contact. These techniques accelerate mixing without compromising the advantages of laminar flow, such as predictable and controlled reactions. Essentially, while diffusion is the primary mixing mechanism, we augment it with channel geometry to achieve the desired results.
Some critics argue that these design tweaks are merely patching over a fundamental limitation rather than solving it. They suggest that relying on such workarounds is a sign that microfluidics might never fully replace traditional methods. What's your view on this skepticism?
I understand the skepticism, but I see these innovations as natural steps in the evolution of any technology. All emerging technologies face initial limitations, and the process of overcoming these is how progress is made. The workarounds we're talking about are not temporary fixes. They are integral to the design philosophy of microfluidics. They enable the precise control needed for applications that traditional methods can't match. In many cases, microfluidic systems aren't intended to replace bulk methods entirely, but to complement them, especially in scenarios where small sample volumes and rapid processing are critical.
Let's move on to real-world applications. One area that's been highlighted repeatedly is the use of lab-on-a-chip devices for medical diagnostics. Can you provide an example of how microfluidics is changing the landscape of healthcare?
Certainly, one prominent example is in the realm of infectious disease diagnostics. During outbreaks, rapid and accurate testing is crucial. Microfluidic lab-on-a-chip devices can analyze a drop of blood or saliva to detect pathogens such as viruses or bacteria. These devices integrate multiple laboratory processes into a single chip, including sample preparation, reaction, and detection. The speed and sensitivity of these systems mean that clinicians can obtain results in minutes rather than hours or days, which is vital for prompt treatment decisions. Additionally, because the devices require only a small sample volume and minimal reagents, they are particularly useful in low-resourcing settings or remote areas where traditional laboratory infrastructure is lacking.
That's a powerful application. It reminds me of how smartphones have revolutionized communication by bringing advanced functionality to a compact form. In a similar way, microfluidics is miniaturizing laboratory tests. Can you share an analogy that might help our listeners understand this better?
Absolutely. Imagine the postal system in a major city. In a conventional system, mail is sorted and delivered by numerous vehicles navigating through busy traffic with the risk of delays and errors. Now, envision a miniature, highly organized postal network where every letter follows a dedicated, unobstructed route with no interference from traffic. In microfluidics, each droplet of fluid is like a letter that moves along precisely defined channels, reaching its destination quickly and efficiently without getting mixed up with other droplets. This organized system ensures that each reaction or measurement occurs under highly controlled conditions, leading to more reliable and rapid outcomes.
That analogy really brings the concept home. Now let's talk about chemical synthesis. There's been a lot of discussion about how microfluidic reactors can offer advantages over traditional batch reactors, especially in producing nanoparticles. What specific benefits do microfluidic systems offer in this domain?
Microfluidic reactors provide exceptional control over reaction parameters such as temperature, pressure, and concentration. In traditional batch reactors, achieving uniformity throughout the reaction volume can be quite challenging. With microfluidic systems, the small reaction volume ensures that heat and mass transfer occur almost instantaneously. This leads to more uniform reaction conditions and consequently products that are highly consistent in size and shape. In the case of nanoparticle synthesis, this level of control is critical. Uniform nanoparticles can have very different physical, chemical, and optical properties compared to those produced in a less controlled environment. The enhanced control can lead to higher yields, improved quality, and even the possibility of discovering new reaction pathways that are not accessible in bulk systems.
While these benefits sound promising, some in the chemistry community remain cautious. They argue that traditional methods still hold the edge when it comes to large-scale production. How do you respond to the notion that microfluidic reactors might be better suited for research rather than industrial manufacturing?
That's a valid concern. Traditional reactors are well established for large-scale production, and they have their own strengths. However, microfluidic reactors excel in scenarios where precision is paramount, such as when producing specialized nanoparticles or when reactions need to be closely monitored and controlled. The scalability challenge is real, but progress is being made through parallelization, using arrays of microfluidic channels operating simultaneously. This approach can potentially match or even exceed the throughput of conventional reactors while maintaining the high level of control that microfluidics offers. So, while microfluidic reactors may not replace batch reactors across the board, they are carving out an important niche in the production of high value or specialized products.
Shifting our focus to another field, environmental monitoring. There are claims that microfluidic devices can detect pollutants at extremely low concentrations. Can you explain how these systems work and what advantages they bring to environmental science?
In environmental monitoring, sensitivity and portability are key. Microfluidic sensors can be designed to detect trace amounts of contaminants such as heavy metals, pesticides, or volatile organic compounds. These devices often incorporate integrated sensors that measure changes in physical or chemical properties, like changes in electrical conductivity, fluorescence, or absorbance when a target analyte is present. Because microfluidic systems use very small sample volumes, they can achieve high sensitivity. Moreover, the compact size of these devices makes them ideal for field deployment where they can provide real-time monitoring of water or air quality. This is particularly important in areas where contamination levels can change rapidly or where traditional laboratory testing is impractical.
It's clear that microfluidics is finding applications across a wide spectrum of fields, but the integration of microfluidics with other advanced technologies like artificial intelligence and nanotechnology seems to be a hot topic. Some critics say it's just a buzzword exercise. What's your take on the integration of these technologies with microfluidics?
The integration is far from mere buzzwords. It represents a natural evolution of the technology. For example, artificial intelligence can be used to analyze the complex data generated by microfluidic devices in real time. This enables adaptive control of the experimental conditions, making the entire process more efficient and reproducible. Nanotechnology, on the other hand, allows for the precise manipulation of nanoscale materials within microfluidic channels. This opens up exciting opportunities in targeted drug delivery and the synthesis of novel nanomaterials. When integrated, these technologies can create multifunctional platforms that are capable of performing tasks that would be extremely challenging or even impossible with traditional methods alone. The synergy between these fields is pushing the boundaries of what we can achieve with microfluidics.
Critics, however, warn that adding layers of complexity might render the systems too fragile for practical use. They argue that over-engineering could lead to devices that are prone to failure. How do you address these concerns?
It's a fair criticism. Increasing complexity does come with risks. However, the key to successful integration is robust design and thorough testing. The goal is not to add complexity for its own sake, but to incorporate advanced functionalities that enhance performance without compromising reliability. Many modern microfluidic systems are being designed with modularity in mind. This means that each component, whether it's the fluidic network, the sensor array, or the data processing unit, can be independently tested and optimized before being integrated into a larger system. Through iterative design and rigorous validation, we can minimize the risk of failure and ensure that the benefits of added complexity outweigh the potential downsides.
Let's take a moment to discuss cellular analysis. There's been a lot of buzz about using microfluidics to study individual cells, especially through organ-on-a-chip technology. How does this approach compare to traditional cell culture methods, and what new insights does it offer?
Traditional cell culture methods typically involve growing cells on flat surfaces, which doesn't accurately replicate the three-dimensional structure and dynamic environment of actual tissues. Microfluidic systems, particularly organ-on-a-chip devices, address this limitation by creating microenvironments that closely mimic in vivo conditions. In these systems, cells are cultured in channels where they experience fluid flow, mechanical stress, and gradients of nutrients and oxygen similar to those in the body. This setup provides a more realistic model for studying cellular behavior, drug responses, and disease progression. For example, liver-on-a-chip systems have been used to study drug metabolism more accurately than conventional cultures. Additionally, microfluidics enables single-cell analysis, allowing researchers to observe the behavior of individual cells rather than relying on average responses from bulk populations. This level of detail is crucial for understanding cellular heterogeneity, which has important implications in cancer research and personalized medicine.
You mentioned earlier an analogy about a postal system to explain the precision of microfluidics. Can you share another analogy or example that illustrates how microfluidics offers a controlled environment for cellular studies?
Certainly, imagine a well-organized library where every book has a designated place and every shelf is arranged by a specific system, making it easy to find any book quickly. In contrast, consider a chaotic pile of books scattered randomly across a room. Finding a particular book would be extremely challenging. Microfluidic devices provide that organized, systematic environment for cells. Each cell, like a book in its specific spot, is isolated and maintained in conditions that replicate its natural habitat. This arrangement allows researchers to study how individual cells react to various stimuli in a controlled, reproducible way, much like how a well-organized library facilitates efficient retrieval and study of information.
Let's turn to the subject of ethics. There's growing concern about the use of microfluidic devices in personalized medicine and genetic analysis, with some suggesting that they could lead to privacy violations or misuse of sensitive data. What ethical considerations should guide the development and use of these technologies?
Ethical considerations are paramount, especially when dealing with technologies that impact human health and personal data. With microfluidic devices used for personalized medicine, the data generated can be very detailed, potentially revealing sensitive information about an individual's health or genetic predispositions. It is essential that strict data protection protocols are implemented and that regulatory frameworks keep pace with technological advances. Researchers, companies, and regulatory bodies must work together to ensure transparency, informed consent, and secure data handling practices. The goal should be to maximize the benefits of rapid diagnostics and personalized treatments while minimizing the risks of data misuse and privacy breaches.
Some critics even suggest that the integration of advanced technologies in microfluidics might inadvertently create new avenues for surveillance or exploitation. Do you think our current regulatory environment is prepared to handle such challenges, or are we entering uncharted territory?
It's a complex issue. The rapid pace of technological innovation often outstrips the development of regulations. While the current regulatory framework is evolving, there is definitely room for improvement. Proactive collaboration between scientists, ethicists, policymakers, and industry stakeholders is essential to create guidelines that are both flexible and robust. This collaborative approach can help ensure that the technology is used responsibly and ethically. It's not that the challenges are insurmountable, but rather that they require ongoing attention and adaptive regulatory measures to keep pace with innovation.
Let's discuss some of the challenges that remain. Despite the significant advances, there are still hurdles to overcome, such as integrating multiple functions into a single chip and ensuring reproducibility across different environments. What are some of the current research directions aimed at solving these problems?
Researchers are exploring several avenues. One approach is the development of modular microfluidic systems that allow different functional units, such as mixing, reaction, and detection, to be optimized independently and then integrated seamlessly. Another focus is on the use of novel materials that offer greater durability and flexibility, which can improve reproducibility and reduce manufacturing costs. Additionally, advances in computational modeling are helping us better understand and predict fluid behavior in complex geometries, which informs the design process. There's also a significant push toward integrating real-time sensing and feedback systems, which can adjust conditions on the fly to maintain optimal performance. This kind of dynamic control is essential for translating laboratory successes into reliable, real-world applications.
Before we wrap up, let's talk about the future of microfluidics. Some experts are extremely optimistic about its potential to revolutionize industries like healthcare, chemical manufacturing, and environmental monitoring. Others remain more cautious. Where do you see the field heading in the next decade, and what realistic milestones should we expect?
In the coming decade, I anticipate several key developments. In healthcare, I expect to see microfluidic devices become a staple in point-of-care diagnostics, delivering rapid, accurate, and multiplex test results. In chemical synthesis, microfluidic reactors will likely be integrated into pilot-scale production processes, especially for high-value or sensitive materials where precision is critical. Additionally, the convergence of microfluidics with artificial intelligence and nanotechnology will lead to more autonomous and intelligent systems. Realistic milestones include the commercialization of robust lab-on-a-chip platforms for routine clinical use, the successful parallelization of microfluidic reactors for industrial applications, and the widespread adoption of portable environmental sensors. While challenges remain, the steady progress in research and development gives me confidence that these goals are within reach.
Steves, you've provided extensive explanations, debated tough points, and shared valuable examples and analogies that really illuminate the intricacies of microfluidics. Before we conclude, do you have a final message for our listeners?
I'd say that the journey of microfluidics is a testament to the power of interdisciplinary collaboration and the persistent quest for precision in science. Whether you're a researcher, a student, or simply someone interested in the future of technology, keep questioning, keep experimenting, and always look for innovative ways to solve complex challenges. The breakthroughs of tomorrow will be built on the foundations we lay today.
And a big thank you to all our listeners for joining us on this in-depth exploration of microfluidics. Remember, every challenge is an opportunity for breakthrough. Stay curious and keep pushing the boundaries of science. Thank you for listening, and we hope you continue to be inspired by the world of microfluidics.
Thank you for the opportunity to share my insights. I look forward to future discussions and debates that will drive this exciting field forward.
Until next time, keep challenging the status quo and striving for excellence. Goodbye, everyone.
Goodbye, and thank you once again for your time and interest.